So in the video you're about to watch, we're going to take a look at some good news and some bad news. Let's start with some bad news. Bad news is that sooner or later, most cells die, which is a problem because living things are made of cells, and if their cells die, then living things would probably die. But the good news is that many cells divide before they die, thus creating new, younger cells and passing on their genetic information. A little more bad news, though. Sometimes cells divide when they shouldn't. These are naughty cells, also known as cancer cells. In this video, on the cell cycle, we're going to take a look at the life of a cell from formation to division. And we'll also take a look at what goes wrong on occasion. As you're watching this video, please note the cool pencil. The cool pencil is there to tell you that a diagram really needs to make it into your notebook. Let's start by looking at the events in the cell cycle. Just as humans go through different events as they grow up, the cell goes through different events. The first phase of a cell's life is interphase, and a cell spends most of its time there. Interphase starts here and goes all the way to this point. So let's start with this cell and follow it along throughout its lifetime. The first part of interphase is G1, where the cell simply grows. Here you can see that the cell has gotten a little bit bigger. The second part of interphase is DNA synthesis, or S. This is when the chromosomes replicate during DNA replication. Here you can see we started with one chromosome in the cell, and now there is a doubled chromosome in the cell. After S phase comes G2. This is where the cell will prepare for mitosis. The cell might make more organelles, it might increase in size, it's going to make sure that everything is in order. Then, if given the signal, the cell will start dividing. And this is mitosis, specifically when the nucleus divides. Here you can see one nucleus with a doubled chromosome, and here is the cell with two nuclei, each containing a chromosome. After the nucleus divides, the rest of the cell, or cytoplasm, divides. So here we've got two separate cells. These two stages together, mitosis and cytokinesis, make up what we call M phase, or mitotic phase. And this is the only part of the cell cycle where the cell will divide. And the end result is two genetically identical daughter cells. And that's super important because the point of mitosis is either for growth, for repair of old or damaged cells, or for asexual reproduction. So we had one cell with one chromosome, and we ended up with two cells, each with one chromosome. Let's take a closer look at S phase, because there's something confusing about the terminology we use here. During S phase, the chromosomes duplicate, as we mentioned. And once the chromosome duplicates, it's held together at the centromere, and it's made up of two sister chromatids. The confusing thing, though, is that this is still just one chromosome made up of two sisters. And then once the cell divides, the two sisters become independent chromosomes. So keep in mind that a duplicated chromosome is just one made of two sisters. Another error that students often make is thinking that this happens during mitosis. This happens during interphase, before mitosis. We couldn't very well divide a cell until we had duplicated the chromosomes. Quick, close look at M phase itself. Here in the cell cycle, we've got these two slivers, mitosis and cytokinesis, but we can divide mitosis itself up into smaller phases. And this will be a quick review because I know you're well familiar with these phases. One thing to remember is that during these three parts of interphase, the DNA is kind of a mess. It looks like this. It's called chromatin. And it's not until the beginning of the first part of mitosis that we start to see the chromosomes. So that first part of mitosis is prophase. And the sign that mitosis has started is that the chromosomes have condensed and are now visible. Some scientists like to put in a phase here called prometaphase. During prometaphase, the nucleus disappears. We can also see that the centrioles with the spindle have moved to opposite ends of the cell. After prometaphase is metaphase, where the chromosomes line up in the middle, and you can see that the spindle is attached to the centromere of each doubled chromosome. Then we've got anaphase, where the spindle separates the sisters. So now the double chromosome has been pulled apart into two identical chromosomes. And then we have telophase and cytokinesis, which kind of run together. We see the two nuclei forming, and these cells are starting to split. Since this is an animal cell, we can see a cleavage furrow. If this were a plant cell, we wouldn't see a cleavage furrow, we'd see a cell plate because of that cell wall. One thing to remember is that even though we divide the cell up into these phases during mitosis, it's really just one continual process, and so these stages are a little bit arbitrary. And again, remember that this process is what's going to make organisms grow, repair their cells, or, if you're a single-celled organism, asexual reproduction. So that was the general cell cycle, and it's easy to think that that's the way it looks in all cells, but that's not the case. Just like different people have different lifespans and do different things during their lives, the same thing is true of cells. For example, some cells never divide. They spend their whole time in parts of interphase. Red blood cells, neurons, and muscle cells are examples of cells that rarely, if ever, divide. So they exist in a non-dividing state known as G0. However, given appropriate signals, sometimes cells in G0 can re-enter the cycle and divide, but not all. And this is one hot area of research, is how to make neurons re-enter the cell cycle so that neurons and nerve damage can be repaired. However, there are some cells that divide frequently. For example, skin cells or embryonic cells. A developing zygote is going to be dividing over and over quite rapidly. Stem cells that you find, for example, in your bone marrow that give rise to red, white blood cells and platelets, those also divide frequently. Mouth cells are constantly repairing and regenerating themselves, stomach cells as well. And then we have some cells that divide every once in a while, maybe every few months or every few years. Liver cells are one example, the pancreas as well. Now let's move on to what controls the cell cycle. What determines whether the cell divides frequently, occasionally, or not at all? Well, throughout the cell cycle, there are checkpoints. There's a checkpoint at G1, a checkpoint at G2, and a checkpoint right before mitosis. And these checkpoints are simply stop-and-go signs. And they're controlled by both signals inside the cell and signals outside the cell. Here's a close-up of what those checkpoints look like. Here, the cell is being given a green light at G1, so it's going to go ahead and move on to S phase. Here, however, the cell is being given a red light, and it's going to enter G0. Maybe this is a red blood cell or a neuron. Molecules are what regulate these checkpoints. Ultimately, the green light or the red light comes down to tiny little molecules. One of the major molecules that regulates the cell cycle is the growth factor. Here you can see that a signaling cell, a cell nearby, is releasing a growth factor, which then moves over to a nearby cell. So these are external signals. Once it's at the neighboring cell, it binds to a protein in the cell membrane, and then it's going to induce a signal, which we'll get to in a second. A couple important examples of growth factors are platelet-derived growth factor. Now this is a mouthful, but it's something that you've experienced all the time. If you ever cut yourself and you've seen blood rush to the side of the cut, and inside that blood, platelets would be clotting to try and prevent further blood loss. Meanwhile, the platelets are also producing growth factors, and those are telling nearby cells to divide by mitosis to replace the cells that were damaged when you cut your finger. Growth hormones are also common growth factors. Another important molecule exists inside the cell, and this is MPF, aka maturation promoting factor. As I mentioned, this will be found inside the cell, and it's actually composed of two smaller proteins called cyclin and CDK. So let's take a look at how these two different types of molecules really work. Starting with the growth factors, again, we can see the signaling cell has released a growth factor. It's bound to the receptor protein, and once it binds to the receptor protein, it can cause a series of changes within the cell. So there might be relay proteins that relay or pass along the signal until the signal gets to the nucleus. And then that signal might tell a certain part of DNA to be transcribed into mRNA, and then the mRNA can be translated into a protein. And that protein might be CDK or cyclin, proteins that we need for cell division. Which leads us to MPF. Once enough cyclin has accumulated, perhaps because of those growth factors, then we'll have cyclin and CDK come together to form MPF. Once they've bound together in this complex of MPF, then the checkpoint can be passed and the cell will enter mitosis. Now, we don't want a cell to be in mitosis all the time, so eventually there will be some enzymes that might come along and break down the cyclin. So then, without cyclin, there's no more MPF and mitosis stops happening. Key points here: CDK and cyclin must bind to form MPF in order for mitosis to happen. And if all is going according to plan, enzymes should break down the cyclin to regulate the cell cycle. Which leads us to cancer, because unfortunately that doesn't always happen in your cells. First of all, what is cancer? Well, it's simply a loss of control over the cell cycle, which leads to excessive cell division. Here you can see cells dividing when really there's no room for them to divide. There's no need to produce more cells in this healthy tissue. And this can lead to a growth of cells called a tumor. Now, some tumors are benign or harmless. They won't grow very large, they can be removed, and nothing happens. However, some tumors become malignant. They continue growing, and then they spread through the blood to other parts of the body. And that's called metastasis, and that's when a person is diagnosed with cancer and is in serious danger. So what makes a normal cell become a cancer cell? Well, if you look at this diagram, here's our normal cell, and here's a malignant cell. The first thing you might see is that there's a lot of steps involved in this process. It doesn't happen overnight. What's really happening as the normal cell becomes malignant is that there is a series of mutations happening in the chromosomes. And those mutations could take months, years, many, many years to accumulate to the point where the cell has become malignant. Let's take a look at a couple genes that will mutate to cause cancer. One major cancer-causing gene is an oncogene. There's also a gene, though, a similar type of gene called a proto-oncogene. This is a good gene. A proto-oncogene might code for a normal relay protein, and this is an important part of the cell cycle. However, if the proto-oncogene mutates, it becomes an oncogene, and this is the bad form of the gene. And this oncogene might code for a deformed protein that's hyperactive. If you've got too much relay protein, you're going to have too much mitosis. So keep in mind, proto-oncogenes are the good guys, oncogenes are the bad guys. And this is why someone who studies cancer is known as an oncologist, because they often are looking at oncogenes. And here we can see the effect of that oncogene. Normally, the proto-oncogene would make a nice little relay protein that sometimes carries the message, but an oncogene will have a hyperactive protein that continues to issue signals on its own. So it's gone rogue and it's causing all sorts of cell division when it has no business doing that. Another very important type of gene is the tumor suppressor gene. A tumor suppressor gene does just that. It's a gene that codes for a protein that might stop cell division. Perhaps it's a protein that breaks down cyclin, or it might be a protein that turns off the transcription and translation of cyclin. And so when these genes are working well, cell division is under control because they're suppressing the formation of tumors. P53 is a very famous tumor suppressor gene. A large portion of cancers are thought to be due to mutations in this gene. Here's what happens if we mutate it. A mutated tumor suppressor gene leads to a defective protein that can no longer control cell division. And now you're going to get tumors. Keep in mind that even though it's genetic changes, genetic mutations leading to cancer, the environment can play a role in causing those mutations. In fact, if you look at the most common types of cancer in the United States, you'll see that there are a lot of environmental factors that cause those mutations to accumulate. Some of the most important ones are tobacco. Everybody knows you shouldn't smoke. Why it's legal, I don't know. Radiation, whether it's UV light or X-ray machines. That's one reason why you're only supposed to get a couple X-rays a year from the dentist. Diet. Fat and salt have been shown indirectly to play a role in cancer. And then there are even viruses that can induce mutations that lead to cancer. So to sum up, during the cell cycle, cells may get to divide by mitosis. This is important for growth, repair, and asexual reproduction, but uncontrolled mitosis can lead to cancer. At this point, please make sure you've got all the diagrams you need in your notes, and you should complete the poll.